Hello everyone, welcome. Today we will be learning about microbes in bioremediation. I am FATHIMA SHAHLA and let's move into our topic. So in this presentation we will be dealing with what is bioremediation, types of bioremediation, and particularly looking into microbes in bioremediation, especially bacteria and its mechanism in the bioremediation of contaminants. So, what is bioremediation? Bioremediation is the process that uses living organisms, primarily microorganisms such as bacteria, fungi, algae, and plants, to remove, detoxify, or degrade environmental pollutants or contaminants into less harmful or less toxic substances such as water, carbon dioxide, and harmless salts. There are different types of bioremediation, and it is the types are uh, bioremediation is classified into different types based on different strategies. So first is in situ and ex situ bioremediation. So this classification is mainly based on where the process of bioremediation is taking place. In case of in situ bioremediation, this is called when the bioremediation is taking place where the original contaminant is there. So the bioremediation, the natural process of bioremediation is in a way in situ bioremediation, where the contaminants are degraded in its own habitat, which is a low cost and less disruptive method, and which can be used for large and deep contaminated plumes. Next one is ex situ bioremediation, in which the contaminants are transferred from the original site to other sites for a faster and more controlled degradation, which is mainly used when the contamination is or the contaminants are less in amount, and the clearing or the degradation of contaminants are urgent in the sites. Next one is aerobic and anaerobic bioremediation. This is basically based on if the organism uses high oxygen or doesn't need oxygen for the process of bioremediation. So in case of aerobic, it will need oxygen or requires oxygen for the degradation of organic compounds such as fuel spills, pesticides, etc. In case of anaerobic bioremediation, the organism does not require oxygen for the degradation of recalcitrant compounds such as chlorinated compounds, PCBs, etc. So next we can differentiate bioremediation on the basis of organism which is involved in the bioremediation, which is microremediation and phytoremediation, in which microremediation is the use of fungi for the degradation of tough pollutants by producing certain enzymes. Pollutants such as heavy metals and other chlorinated compounds are degraded by microremediation. And in case of phytoremediation, it is the use of plants for the remediation of contaminants from a site. It is usually used in large, low or medium contaminated sites. Next we are looking into microbes in bioremediation. So basically microbes which are involved in bioremediation are bacteria, fungi and algae. In this video we will be more discussing on the bacteria which are involved in bioremediation. So bacterial bioremediation. Bacterial remediation is simply the use of bacteria or bacteria converting or degrading organic pollutants or recalcitrant pollutants into less harmful or less toxic compounds such as carbon dioxide, water, etc. This can be bacterial bioremediation can be further divided into aerobic and anaerobic based on the requirement of oxygen. So aerobic bioremediation. In case of aerobic bioremediation, the organisms or the bacteria will use the organic compounds or organic pollutants or the contaminants as a source of electron for the process of energy production and synthesis of carbon compounds for the cellular building. So the final acceptor of electron in the electron transport chain will be oxygen. Bacteria produce various oxygenase enzymes in order to make the stable hydrocarbons functional, and thus the bacteria will degrade the hydrocarbons and make substrate uh, which can be incorporated into central metabolic pathways such as TCA, and then further this compounds or substrates are converted into energy carriers, and thus energy can be attained from the organic pollutants or other contaminants. So aerobic bacteria uses oxygen for twice in the whole process. First, for the activation of the stable hydrocarbon in the initial attack, and in the last stage of the respiratory pathway for the final acceptor, as the final acceptor of the electron. So some of the aerobic bacteria also produce biosurfactants, which makes the stable hydrocarbons available for the or pollutants available for the bacteria to easily metabolize them. Mechanism of aerobic bioremediation is as follows. So first is initial attack. In initial attack, it is basically for making the stable hydrocarbon or organic compounds, pollutants, into a functional one so that the bacteria can use it for the metabolism. So the process is like this. The organisms or the bacteria will produce enzymes such as oxygenases, which will act on the stable organic compounds or pollutants, making them chemically unstable and soluble in water. This enzyme oxygenases are of two types: monooxygenases and dioxygenases. This is monooxygenases means they add one atom of organic oxygen molecule into the organic compounds to make them unstable and soluble water compounds. For example, the monooxygenases add an atom of oxygen into alkane to make them a primary alcohol. In dioxygenases, they add both atoms of oxygen from the oxygen molecule into hydrocarbons. This dioxygens mainly act on aromatic compounds. Next step is degradation by peripheral pathway. So the initially oxidized compound is transferred or passed through series of enzymatic reaction to break down this complex molecule into simpler substrates such as acetyl-CoA, succinate, pyruvate, etc., which are involved in the assimilative cycle such as TCA. This substrate will further enter into the TCA cycle or tricarboxylic acid cycle, and thus there is two basic things that's happening in the TCA cycle, that is energy production and biosynthetic precursor generation. In energy production, the completely oxidized carbon skeleton from the pollutants will generate high energy electron carriers such as NADH, FADH2 and ATP or GTP. In case of biosynthetic precursor generation, TCA cycle provides key intermediate for building cellular components such as amino acids and nucleic acids. And the final part where the energy carriers or the high energy electron carriers formed in the central metabolism TCA cycle are converted into usable chemical ATP such as usable chemical energy, that is ATP. So the NADH and FADH2 will donate electrons to the electron transport chain in the cell membrane, and this movement of electrons through the electron transport chain will stimulate the pumping of protons from across the membrane, creating a proton gradient. The energy from this will drive the ATP synthase to produce ATP. And at last or finally, the electron in the electron transport chain will be accepted by oxygen, and a proton will combine along with them to form water. Thus, the carbon from the hydrocarbon is fully oxidized to carbon dioxide. Next, we are looking at some examples of aerobic bacteria which are involved in the degradation of certain classes of pollutants. So, bacteria such as Pseudomonas, Rhodococcus, alkanivorous oxygenase are involved in the degradation of petroleum hydrocarbons. Mycobacterium, Methylocyanus, Pseudomonas are involved in the degradation of chlorinated solvents. Comomonas and Rhodococcus are involved in the degradation of PCBs. And Sphingobiums and Burkholderia are involved in the degradation of several pesticides. Bacterial species such as Pseudomonas and Enterobacter are involved in the degradation of several explosives. So next we are moving on to the topic anaerobic bioremediation. So in anaerobic bioremediation, the final electron acceptor will be any other electron acceptor other than oxygen. So it can be a nitrate, a sulfate, or even a carbon dioxide, etc. So this anaerobic bioremediation usually takes place in anoxic conditions such as aquifers, sediments, waterlogged soil, and landfills. Based on the key metabolic, there are different metabolic strategies followed by anaerobic bioremediation such as anaerobic respiration, fermentation, and syntrophy. There are also based on the final electron acceptor in anaerobic respiration, anaerobic bioremediation can be further divided into mechanisms can be further divided into nitrate reduction, metal reduction, sulfate reduction, and methanogenesis. The last and final one is organohalide respiration, in which anaerobic bioremediation is attained by using this or pollutants as final acceptor of electron rather than using them as a donor or source of electron or so as a food source. So first we are going to look into different metabolic strategies, that is anaerobic respiration, fermentation, and syntrophy. Anaerobic respiration, we have already discussed that the contaminants is oxidized and the electrons are passed into the electron transport chain and further will be combined with a electron acceptor other than oxygen, thus synthesizing ATP molecules. In case of fermentation, the contaminants will act both as an electron donor and an electron acceptor, but this is energetically less favorable since it have less energy production. Then next is syntrophy. In this, the mutualistic partnership where the degradation of compounds is only thermodynamically feasible if the reaction products are kept at very low concentration by a partner organism. So the anaerobic microorganisms will attack the pollutants and degrade them, and finally the whole product which is accumulating in the environment will be removed by a mutualistic partner organisms like methanotrophs, so that the thermodynamic feasibility is attained. First of all, nitrate, so we are going into further into anaerobic respiration by different terminal electron acceptors. First we are looking into nitrate reduction, in which the electron acceptor is nitrate. So the pollutants are considered as electron donors. So electrons are taken from the organic pollutants or contaminants, and then these electrons are used to convert or to reduce nitrate into atomic nitrogen, which is less harmful. So the contaminants degraded by nitrate reduction are benzene, toluene, ethyl benzene, xylenes, and alkanes and phenols. These are some of the examples of the contaminants which can be degraded by nitrate reduction process. Key bacteria involved are Paracoccus denitrificans, Pseudomonas schützli, Thiobacillus denitrificans. So in case of nitrate reduction, the pollutants will be metabolically converted from the complex substance into a smaller substance, and the electrons from this compounds are given to the ETC. Further, these electrons will combine with the nitrate molecule to reduce it into atomic nitrogen. This nitrate is converted into nitrogen gas and this process is known as denitrification, which provides highest yield of energy after aerobic respiration. Next one is metallic reduction. So in metallic reduction, which is in which the metals will act as the final electron acceptor. Metals such as manganese, iron are involved in this. This bacteria use metals for the production of energy rather than assimilating the metals in them. So this microorganisms will have specialized cytochromes in their membrane, outer membrane, that will directly transfer electrons into insoluble oxides, iron and manganese oxide. They can also use soluble humic acid as electron shuttles. The key bacteria or some of the bacteria which are involved in metallic reduction of are Geobacter metallireducens and Schwannella onidensis. Some of the contaminants which can be degraded by this method are aromatic compounds such as toluene, phenols, picrosol, and then acetate. Even complete oxidation of fatty acid to carbon dioxide can also be attained by metal reduction. So, in metal reduction process, the organisms can immobilize toxic metals by reducing soluble metals into insoluble and mobile contaminants into immobile states. Next one is sulfate reducing or sulfate reduction. The sulfate reducing bacteria or SRB such as desulfovibrio, desulfobacter, etc. will use sulfate as a final acceptor of electron from the ETC. So, in case of sulfate reduction, there are majorly two stages, in which first stage is activation, in which a sulfate, which is an already stable molecule, is converted into adenosine 5-sulfate by the help of enzyme ATP sulfurylase is done in the first stage of activation. Thus, the activated adenosine 5-phosphorsulfate APS will further acted by APS reductase to convert APS to sulfite. This sulfate is then converted into hydrogen sulfate by a series of 6 electron reduction. So the electrons for the process of the reduction of sulfate into hydrogen sulfate is obtained from the contaminants or the organic pollutants. Thus, the contaminants are used up by the bacteria. And also, sulfate reducing bacteria uses fumarate addition method to attack stable hydrophobic molecules such as benzene and gasoline, etc. So this is the pictorial representation of fumaric acid addition used by sulfate reducing bacteria. When we in degradation of toluene is incorporated into across the double bond of the fumaric acid, thus forming benzyl succinic acid with the help of the enzyme benzyl succinate synthase. So this benzyl succinic acid is further undergo series of oxidation similar to beta oxidation of fatty acids to form benzyl CoA. This benzyl CoA is a central intermediate in anaerobic aromatic metabolism, and thus further the ring is reduced and cleaved and eventually will be feeded into the central metabolic pathways to produce energy and carbon for growth. Next one is methogenesis. So in aerobic or in methogenesis, the final electron acceptor will be a carbon dioxide, and the resulting product which is formed will be methane. Methanogens will not initiate the attack on pollution, but they actually helps in the maintaining of thermodynamic drive necessary for the complete mineralization of the complex organic pollutants. So they act as a electron sink so that the products won't accumulate in the environment of biodegrading environment, so the other organisms can actually work well for better bioremediation of the contaminants. Next one is organohalide respiration. This is as an anaerobic process, but it different from another anaerobic processes in a way that in this the chlorinated solvents will act as a terminal electron acceptor rather than using them as a food source. So the bacterium oxidizes a simple electron donor and pass the electron through ETC or electron transport chain, and the electron is passed to the chlorinated compounds. The chlorinated compounds along with the electron and proton by the action of reductive dehalogenase removes chloride from the pollutant and replace it with a hydrogen atom. Thus RCl plus 2 electron plus hydrogen gives a dechlorinated compound, pollutant and a free chlorine. The dechlorination happens in a series of dechlorination pathway. Thus, the tetrachloroethene is converted into trichloroethene by the action of reductive dehalogenase and further into cis-1,2-DCE and then to vinyl chloride and ethene. In case of cis-1,2-DCE, this is a compound or the degradation of cis-1,2-DCE is not attained by every organism. So this can be called as a stall point. Whereas vinyl chloride is termed as a carcinogen. So the accumulation of vinyl chloride is not good in environment. So the conversion of vinyl chloride to ethene is a critical step, and this is helped by the enzyme vinyl chloride reductase. Thus ethene is less harmful compound is formed. Next is dihalococoids. Dihalococoids are obligate organohalide respirators which require chlorinated compounds to grow. They have several reductive dehalogenase enzymes encoded in their genome so that the complete degradation of organohalides into ethane can be attained by them. These are very helpful in successful destruction of carcinogenic vinyl chloride from the environment. This is the pictorial representation of organohalide respiration. So in this, the lower part shows the aerobic metabolic oxidation and co-metabolic oxidation, whereas the upper part where the arrows full arrows are shown is the anaerobic reductive dehalogenation. So in anaerobic reductive dehalogenation, with the help of reductive dehalogenase enzyme, the PCE is converted into TCE, then further into cis-DCE, then to vinyl chloride and then ethene, which is a complete degradation of organohalide compound. Whereas in case of aerobic metabolic and co-metabolic oxidation, the intermediate compounds from the anaerobic reductive dehalogenation or the compounds such as TCE, cis-DCE, vinyl chloride, ethene are converted into phenols alcohol epoxides by monoxygenase or dioxygenase activity, and thus forming TCE epoxide, which is an intermediate in the aerobic metabolic or co-metabolic oxidation. So this epoxide is a highly reactive and toxic substance. So formation of this will affect the destruction, or this will cause the destruction of microorganism or bacteria, and also the accumulation of toxic substance in the environment. This will aerobic oxidation and co-metabolism of halochlorides or chloroethanes, or we can say organohalides, will not be completely attained, or the complete mineralization of organohalides can be attained by an aerobic process or aerobic oxidation, whereas anaerobic reductive dehalogenation process will help us to completely degrade these organohalides into less harmful ethene. In conclusion, bioremediation gives us the idea of transform and destroy rather than the conventional idea of dig and dump. This offers a sustainable, cost-effective and natural solution to environmental contamination. By understanding specific bacteria and their mechanisms, we can understand bacteria more and strategically manage these bacteria to attain a efficient bioremediation process. So, rather than taking aerobic versus anaerobic, using a synergy of aerobic and anaerobic will be very useful for bioremediating many kind of pollutants in our environment. In order to increase bioremediation, we can use two methods such as biostimulation and bioaugmentation. Biostimulation is we provide missing nutrients or electron donors to supercharge the natural abilities of the organism present in the environment to have an efficient bioremediation. Wherein bioaugmentation, we will be adding specialized cultures like dihalococoids to fill functional gaps and ensure complete degradation. So, I will be concluding my presentation. Thank you so much for your attention. The next two microorganisms, fungi and algae, will be discussed in the next video by my friend Bala. Thank you so much.